Welcome back, everyone. So I'd like to continue our discussion on chromosomal mutations or chromosomal variants that lead to phenotypic consequences of the organism. So we've talked about a number of variants to this point. We've talked about the aneuploids and the polyploids. What I want to discuss now are structural abnormalities that are found associated with chromosomes that, again, when they occur, can cause phenotypic results or phenotypic consequences. And again, generally, these phenotypic consequences are going to be negative. So this is just a summary of the different types. We're not going to talk about all of them within this lecture, but we're going to cover a few of them. So the first one is the deletion up here, the upper left, duplications, inversions, and then two different types of translocations, a reciprocal and a non-reciprocal type of translocation. So again, first of all, what I'm going to do with each of these different types of structural abnormalities associated with chromosomes is, provide some definitions of terminology. Secondly, describe mechanisms that will create these abnormal chromosomes, and then give you some examples of phenotypic consequences that result. So let's start with the deletions. So again, a deleted version of a chromosome, again, the normal chromosome that resides in the genome of a particular species. The deleted version is demonstrated here. So again, if you think of these different letter designations along the length of this chromosome as being either blocks of genes or individual genes, there's, as I've said before, there's a very characteristic set of genes that are associated with any given normal chromosome within an organism's genome. And there's a very characteristic order in which those genes reside within that chromosome. So a deleted version is simply you're missing a portion of the original chromosome and all of the genetic information within that region. So in this case, it's the D region. The deleted version of the chromosome no longer carries any of that genetic information within that D region of the chromosome. And that can be, the deletion can encompass a part of a gene, it can encompass an entire gene, or it can encompass multiple genes. In other words, these deletions can be of variable size. Okay, so a duplication is the opposite. So again, if this represents the normal chromosome on the left, A, B, C, D, E, F, G, again, a normal set of genes and a normal order to those genes. Here's a duplicated version of the same chromosome. So actually what you see for this particular example is you're duplicated for the BC region. Okay, BC and then BC, again, duplicated tandemly on that chromosome, that variant chromosome. So actually what that means is that, again, whether it's a single gene or multiple genes within the BC region, on a single chromosome, you now have multiple copies of that gene or those genes. So that's unusual. Again, normally you're supposed to have a single copy of a gene on a single chromosome. And then within a diploid genome, you would have two copies of the gene, one on each member of the homologous pair of chromosomes. So you can think of duplications and deletions as partial aneuploids. Again, remember with aneuploids, you were either missing entire chromosomes or you had extra copies of entire chromosomes. In this case, you're either deleted, deficient for a portion of a single chromosome, or you carry extra genetic information within it, the duplicated version of the chromosome. So again, as I said, you can think of duplications and deletions as partial aneuploids. And we already know what the consequences of aneuploidy is. There are significant phenotypic consequences that result. So in addition to duplications and deletions, you can have what are called inversions. So inversions are a little bit different. In this case, you don't lose information as you do with a deletion. You don't gain information as you do with a duplication, but instead you simply reorganize the information, the genetic information within the chromosome. You change the linear order of genes. So here's again the normal chromosome, A, B, C, D, E, F, G. And here's an inverted derivative of that normal chromosome, A, D, C, B, E, F, G. So you're inverted for the B, C, D region. So again, you don't lose information, you don't gain information, you just reorganize it. You change the linear order of genes on the chromosome. And believe it or not, that there's at least the potential where that type of inversion can cause phenotypic abnormalities. Okay, so then lastly, I said a fourth type of structural variant for chromosomes are what we call translocations, and there's two different types. Here's one diagram representing a non-reciprocal type translocation. So in this case, what you do is you exchange information. For a translocation in general, you exchange genetic information between two non-homologous chromosomes. So here are the two normal versions of these two non-homologous chromosomes within an organism's genome, to the left in this diagram. And then on the right, you can see a portion of the blue chromosome has been translocated to the brown or orange chromosome, so the AB region. So again, so it's non-reciprocal. The direction of transfer of genetic information is from one chromosome to the other, from one non-homologous chromosome to another. So again, you haven't lost information, you haven't gained information, you simply reorganized it within the genome. And then the reciprocal translocation is where the exchange goes both ways. So here's the two original chromosomes, non-homologous chromosomes within the organism's genome. And here you can see you've reciprocally swapped genetic information between these two non-homologous chromosomes. So now you have two hybrid chromosomes, if you want to look at it that way. So you see the HIJ region, originally present here, is now on the blue chromosome, and the AB region, again originally present here, is now present on the brown chromosome. So reciprocal or non-reciprocal, both of these types of translocations can have or result in phenotypic consequences, and we'll talk about them as we go along. So what I want to do is, again, take each one of these one by one, and again, talk a little bit about mechanisms that will produce these abnormal chromosomes, and then give you examples of phenotypic consequences. So let's look at the deletions first. So deletions, what I should say is that all of these structurally abnormal chromosomes are rare occurrences in any organism. Okay, again, and one of the reasons for that is the mechanisms that give rise to these chromosomal abnormalities are rarely acting, or they're caused because of random errors or random damage or random mistakes. So this is certainly true with deletions. So how do you create a deleted version of a chromosome? So what you have to do is you have to break the chromosome. So here's an original chromosome up here at the top. This blue chromosome, A, B, C, D, E, F, is in terms of the linear order of genes. Say those represent genes. Those letters represent genes on the chromosome. So then you get a break. So how do you get a break in a chromosome? Again, remember, that's one long molecule of DNA that runs from end to end, and then it's embedded in this matrix of proteins. That's what the chromosome is. So how do you break the chromosome? Well, you break the DNA molecule. You break the chemical bonds such that you fragment the DNA molecule. So that break can occur, totally randomly, either through exposure to different types of chemicals, some sort of an environmental exposure, or radiation. Different forms of radiation, ionizing forms of radiation, have high enough energy that they can actually break chemical bonds, and in this case, break the DNA molecule into fragments. So you fragment a chromosome. So if that's the case, you'd end up with these two fragments over here to the right after that break has occurred. Okay, so what's going to happen is that, the cell actually has repair machinery. We're not going to talk about the details of the repair machinery, but once that type of damage occurs, there's repair machinery that allows the cell to put the chromosome back together again, try to repair the damage. But it doesn't always work. And if it doesn't always work, what's going to happen? Well, you have these two fragments. You have now this chromosome that is deleted for the A region because of that break, and then you have this fragment of the chromosome that carries the A region or the A gene. So what's going to happen if that cell tries to divide mitotically, let's say mitotically? Well, what's going to happen is the that A containing fragment does not contain a centromere, and therefore it cannot attach itself to a spindle apparatus, and therefore cannot segregate normally during the mitotic anaphase. So actually what's going to happen to that fragment is it's going to be left out in the cytoplasm, at the end, by the completion of the mitotic event, telophase, and it's going to be degraded and lost. So the end result is that in the nucleus, you retain this chromosome with the fragment with the centromere, and it's now deleted. It's deleted for that A region. And again, it could continue then to be propagated through subsequent rounds of cell division in that deleted form. So obviously, these are not common events. As I said, it requires some sort of an environmental exposure that leads to chromosome damage that then would produce the deleted version of the chromosome. And again, remember, all of this is what I'm describing are events that would be taking place in somatic, non-reproductive. This could occur in reproductive tissues as well, but that would be would be a rare event. So alternatively, another way that you can create deleted versions of chromosomes is through recombination errors. So we've talked about recombination. Recombination is supposed to take place during a meiotic event, during early prophase 1. Okay, well, and there's a whole machinery that is responsible for giving rise to these recombination events. So, it's not a simple process. Okay, so in any event, sometimes that recombination machinery can go haywire. It functions abnormally. And say it were to become activated in mitotic or somatic, mitotically dividing cells. Well, again, what potentially can happen is you can get a point of overlap within the arm, as they're representing here in this diagram, the arm of a single chromosome. So that's a potential point for a recombination event, similar to a chiasma that forms within the tetrad during meiosis. So that point of overlap, if the recombination machinery becomes activated, can actually lead to a recombination event at that point of overlap within the single chromosome. So what is that going to lead to? Depending on how the recombination takes place, it can lead to a deleted version of a chromosome. So in this case, you'd become deleted for the DC region. And actually, what it would give rise to is a ring chromosome that carries that DC region or those DC genes. So again, if this is occurring mitotically, what's going to happen if this cell tries to, or in a somatic cell, if this cell tries to divide, what's going to happen to that ring? Well, it's going to be lost because it doesn't have a centromere. It can't segregate properly during a mitotic event. So it will be lost, and what you're left over with in the nucleus of the cell, the daughter cells, is this deleted version of the original chromosome. Okay, so these obviously are rare events, but they do occur. Okay, these two mechanisms, either recombination errors or random fragmentation of chromosomes through environmental exposures to chemicals and different forms of radiation, can give rise to deleted chromosomes. Here's a more common mechanism and a more significant. I mean, I say common; it's still rare, but relative to the two previous mechanisms I've just described, it's more common. So again, and this is more significant, or this mechanism is more significant because of the fact that it occurs meiotically, during a meiotic event, and therefore the deleted chromosome, instead of being propagated through subsequent mitotic cell divisions, um, within the individual, the somatic cells within the individual, you produce a gamete that carries a deleted version of the chromosome and therefore can be transmitted to offspring. And all of the cells of the offspring would then be generated, through mitotic events after zygote production, and therefore all of the cells of the offspring would carry the deleted chromosome, 100% of the somatic cells. So what goes wrong during meiosis to give rise to a deleted chromosome? So it's recombination errors again. So again, what's supposed to happen during prophase 1 is replicated homologous chromosomes pair up with each other to form a tetrad. And the alignment is supposed to be that homologous regions of each chromosome within the tetrad, homologous regions are supposed to pair up with each other. So A is supposed to pair with A, B with B, C with C, D with D, E with E, and F with F. And that happens most of the time. But occasionally you can get this misalignment, as shown in this diagram here. So if you have a misformed or a misaligned tetrad, which then subsequently undergoes a recombination event, as is being shown here, you're going to produce, as the products of recombination, these two chromatids here, 2 and 3, both of them are going to be abnormal because of that misaligned tetrad. Okay, and the end products are shown over here. Chromatid 2 now, or the one labeled chromatid 2, is now deleted for the C region. And chromatid 3, which will end up in a gamete, is now duplicated for the C region. So this process of unequal crossing over, what's called unequal crossing over, that occurs because of an error in recombination during a meiotic event, gives rise to both duplicated and deleted versions of chromosomes, which can then be transmitted to offspring. So a much more significant mechanism for creating either types of these chromosome abnormalities than the previous two mechanisms that I've described. So then the question is okay, so when you produce a deleted version of the chromosome, let's get back and just focus on deleted chromosomes. What are the phenotypic consequences that result? Or are there phenotypic consequences that will result? So this is a pretty straightforward example, and it's not too dramatic, but at least it gives you the idea. So there's a gene in the Drosophila, the fruit fly genome. It's called the Notch gene. That is a gene that contributes to wing formation. Okay, and so this is what a normal wing looks like when you have two copies of the Notch gene, one on each member of a homologous pair of chromosomes within the Drosophila genome. So normal two copies on that chromosome. However, if you're heterozygous, so that you have one normal chromosome that carries the Notch gene, and the second homologous partner to that chromosome is deleted for the Notch gene, what results phenotypically? Well, the wing is abnormal. It obtains, because of abnormal development, a little notch being formed at the base of the wing, or back at the back end of the wing. Okay, so again, carrying a single copy of the Notch gene, because one chromosome is deleted for the Notch gene, gives rise to this phenotypic abnormality. Okay, that may not be too dramatic, but at least you get the point, is that phenotypes can be altered when you carry deleted versions of chromosomes. So here are some more dramatic examples of that. We'll just give you a quick run through of this table here. These are actually genetic conditions that occur in humans because of the presence of deleted versions of human chromosomes. The first example of this is Wolff-Hirschhorn syndrome. This is caused by a deletion of the tip of the short arm of human chromosome 4. So again, remember the convention is when you have submetacentric or acrocentric chromosomes in a human genome, you call the smaller of the two arms the P arm and the longer of the two arms the Q arm. So that's chromosome 4. It's submetacentric. You have a short arm and you have a long arm. The short arm is the P arm, and the tip of that short arm is deleted in this to give rise to this particular condition or this particular syndrome. So what that means is that the individuals that suffer from this condition, they're going to be heterozygous for one normal chromosome 4 and one deleted version of chromosome 4 for that tip of that short arm. And therefore, they're only going to carry one copy of all of the genes that fall within the region of that deletion. So what are the consequences? Well, they're pretty significant. There's growth retardation, heart malformations, cleft palate, and viability is compromised. 1 in 3, 30% die within 2 years post-birth. So here's an example of a child that has Wolff-Hirschhorn syndrome, heterozygous for that deleted version of chromosome 4. I don't know how obvious it is here, but there's a repaired cleft lip. The eyes are more widely spaced than is typical average healthy individual. And again, this individual suffers from mental retardation as well. So here's what the deleted version of the chromosome looks like. Here's the normal human chromosome 4 on the left. Here's the centromere right here. So this is the short arm. This is the long arm, the Q arm. There's the tip of the long arm, and you see it's missing over here. So this is the deleted version of human chromosome 4 that gives rise to Wolff-Hirschhorn syndrome. So you're missing just that small tip and all of the genetic information contained within that region of human chromosome 4. So that probably represents, overall maybe 1% or less of the entire human genome. And yet, being heterozygous for that deleted version of that chromosome, you suffer all of these consequences that are associated with Wolff-Hirschhorn syndrome. Okay. So I should point out is that it would be, first of all, these deleted chromosomes are pretty rare. So it'd be pretty rare that you'd be homozygous for the deleted version of the chromosome. Just in a population, it'd be extremely rare that you would produce an individual that would be homozygous for the deleted version of the chromosome. But even if you did, that would be a non-viable condition. Because there's, again, you create a genetic imbalance. Get back to this theme that I've talked about previously with aneuploids and polyploids, is that you'd be missing both copies. If you were homozygous for the deletion, you'd be missing both copies of the genes that fall within that region of the deletion. So you create a genetic imbalance again that is going to be a non-viable state. Okay, let's go back to the table. So here's another syndrome, Cri-du-Chat. It's the same idea. This is the tip of the short arm of chromosome 5. Again, if you are heterozygous for that deleted version of chromosome 5, you suffer all of the effects, all of the different conditions associated with this particular syndrome. So infants have the name is derived from a very characteristic cat-like cry that infants display. There are facial abnormalities and severe mental retardation, again, that's associated with these individuals. So here's a karyotype of an individual that suffers from Cri-du-Chat syndrome. Here's chromosome 5. Here's the normal chromosome. This is the short arm here. You can see you're missing the short arm, the tip of the short arm on this second copy of chromosome 5. This is the deleted version. And simply by missing that portion of chromosome 5, again, which represents probably 1% or less of the entire human genome, you suffer all of the consequences of this particular genetic condition. So again, the eyes have an almond shape to them. The teeth form abnormally. Nails, fingernails form abnormally, and again, severe mental retardation associated with this condition. Here's another example. Here's normal chromosome 5. Again, there's the short arm, the P arm. Here's the deleted version, represented schematically over here, represented in the karyotype down here. And here's the girl with this particular karyotype. So again, you can see the almond-shaped eyes, teeth form abnormally, mental retardation, and there's a very characteristic hand-wringing behavior that's associated with this condition as well. So here are two examples. There's something called Wilms tumor and a form of retinoblastoma. These are both cancers. Wilms tumor affects or forms tumors in the urogenital tract of individuals, and retinoblastoma is a form of eye cancer. So again, both of these conditions can be caused by the individual being heterozygous for a deleted version of a human chromosome. In the case of Wilms tumor, it's the tip of the long arm of chromosome 11 that is deleted, and for retinoblastoma, it would be the tip of the long arm of chromosome 13. Okay, so again, it would be very rare that these individuals would be homozygous for that deletion, probably never really occur. And even if it did, those would be lethal conditions. Okay, so I do want to mention one complementary syndromes, a little bit unusual, and we can talk more about why this is later on. It's a more complicated genetic system, but there's something called Prader-Willi syndrome and Angelman syndrome. And actually, the two syndromes are caused by the same deletion, or very closely related deletions on human chromosome 15. And the majority of those individuals that suffer from either Prader-Willi or Angelman syndrome have an internal deletion. So all of these previous examples I've given you are what we call terminal deletions, the tips of the arms of the chromosome. So an internal or interstitial deletion would be internal to the arm, so it requires 2 breakpoints instead of 1 to lose the region of the chromosome, the deleted region of the chromosome. So for Prader-Willi and Angelman, that interstitial deletion occurs near the centromere on chromosome 15, the long arm of chromosome 15. So here's a child that suffers from Angelman syndrome, and here's an individual that suffers from Prader-Willi. So the outcomes are, there's related consequences that occur for either of these two syndromes, but they're also different in many ways as well. What you see, again, very commonly with Prader-Willi is, first of all, they can survive into adulthood, but they have a compulsive eating behavior that very commonly leads to obesity in these individuals. And you can see Angelman, those that suffer from Angelman syndrome generally are weak and frail and not particularly healthy, and have a much more difficult time to survive and grow into adulthood. And you go back to the table here. So, Prader-Willi, infants weak, slow growth, obesity, compulsive eating, Angelman syndrome, developmental delays. So again, as a child, infant, early childhood, very slow growth and development, and significant mental retardation associated with these individuals as well. So if the deletion covers a similar region of chromosome 15, why do you get such different outcomes? So we'll talk about that in a second. So I do want to point out is that this diagram at the bottom here represents chromosome 15. Here's the centromere to the left, and then this is the long arm, the Q arm of chromosome 15 in the region of the centromere. This is where the deletion occurs for both Prader-Willi syndrome and Angelman syndrome. And so you can see that we have a pretty detailed genetic map. Multiple genes fall within that region of the deletion. Okay, so then the question is, well, why do you get the different outcomes? Well, it depends on which parent transmits the deleted version of the chromosome that determines whether or not you suffer from Angelman syndrome or Prader-Willi syndrome. So again, here's chromosome 15. This is a different depiction than what I just showed you. Here's the centromere. Here's the region of the deletion on the long arm, requiring those 2 breakpoints. Well, if that deleted version of the chromosome is transmitted through the father, through the paternal gamete, you'll suffer from Prader-Willi syndrome. Alternatively, if the deleted version of the chromosome is transmitted through the maternal parent, the mother, you're going to suffer from Angelman syndrome. And the reason for that is a little bit complicated. If you're familiar with a phenomenon known as genetic imprinting, it's that phenomenon that plays a role in this unusual situation where the particular condition that is going to result depends on which parent transmits the abnormal chromosome. Okay, so those are the deletions. And again, giving you an idea of mechanisms that generate deleted versions of chromosomes, and then some of the consequences, phenotypic consequences that can result by carrying a deleted version of a chromosome within your cells. Okay, so let's talk a little bit about the duplications. So what are the mechanisms that cause duplications? Well, again, there's a couple of different mechanisms. In this case, what do we know about chromosome replication? Well, we know that replication of genetic information takes place during the S phase of the cell cycle, either mitotically or meiotically. So actually, errors in DNA replication during the S phase of the cell cycle can give rise to duplicated versions of chromosomes. So in other words, the replication machinery goes haywire and makes a mistake. So the end result of this abnormal replication is that you create a duplicated version of the original chromosome. So pretty rare, but it does occur. Okay, the second mechanism, which is a little bit more frequent, still rare, but a little bit more frequent than replication errors, is again this unequal crossing over that I've described already. And again, in this case, what's going to happen is you're going to create an abnormal gamete, in other words, a gamete carrying a duplicated version of the original chromosome, and for this example, duplicated for the C region, and then that's going to be transmitted to progeny, to an offspring. And then all of the cells that comprise the body of that offspring are going to be carrying the duplicated version of the original chromosome. Okay, so this is really the main mechanism, this unequal crossing over to create duplicated versions of chromosomes. So what I want to give you examples of then are, the phenotypic consequences that are going to result from these duplications. There's actually three different types of duplications. There are what we call micro duplications, which are less than gene size. You have gene size duplications. So in other words, for this previous example, say C represents an individual gene, this chromosome would then be duplicated for that individual gene. Alternatively, you can have larger duplications that encompass large regions of chromosomes, such that that C region might contain, 10 or 20 genes. So you're duplicated then in that abnormal chromosome for those 10 or 20 genes. So these are three different types of duplications that I want to talk about with regard to phenotypic consequences that can result. So let's look at the micro duplications first. And again, the mechanism that are going to produce these micro duplications are going to be primarily not unequal crossing over, but replication errors during the S phase of the cell cycle. So in other words, these are less than gene size duplications. So again, remember the gene is represented as a DNA molecule, a DNA sequence. And so here's an example of a microduplication. These are what we call trinucleotide repeats. And when these types of microduplications occur, you create, what we call trinucleotide repeat disorders. And here's a whole table of different genetic disorders that result from this type of microduplication, this trinucleotide repeat duplication error. So let me just pick one example. So we've talked about Huntington disease before, again, as a genetic disorder that is caused by a dominant allele. And so what makes the allele defective in such a way, this dominant allele defective in such a way that it gives rise to all of the conditions associated with Huntington disease? What happens is that within the gene sequence, there's a 3-nucleotide sequence, CAG, and through multiple rounds of replication errors, that trinucleotide sequence is replicated and duplicated. And so the numbers of that trinucleotide repeat increase through many rounds of replication. So in the normal allele that a normal healthy individual carries for this particular gene, this gene responsible for Huntington disease, is the normal gene. The normal allele carries anywhere between 9 to 37 tandem copies of this CAG sequence. But because of replication errors that lead to duplication, you can create the defective dominant allele that within it contains greater than 37 tandem repeats of that 3-nucleotide sequence. And so that's the defective form of the gene, the defective allele, that causes disease in a dominant manner. So how do you get those extra copies again of that 3-nucleotide sequence? It's because of replication errors. So there are many examples of this, Fragile X syndrome, Jacobson syndrome. You can read the list down here. There's actually many more examples of this. This is just a partial table or a partial list of these trinucleotide repeat disorders. So microduplications give rise to allelic variants for individual genes that the allelic variant that contains the duplicated region, in this case, this 3-nucleotide sequence, that it renders the allele defective or non-functional such that disease results. So this is, Fragile X was in that list there. So this occurs on the X chromosome. It gives rise to a form of mental retardation. And again, within this region here, here's the normal chromosome. This is the tip of the long arm of the X chromosome of humans. What happens within that region is you get this trinucleotide repeat duplication occurring beyond a threshold level such that you create this abnormal chromosome that it looks like it's called Fragile X because it looks like you can see over here in this uh, karyotype analysis here, looks like the tip is about to fall off of the chromosome because of the presence now of the duplication within that region. Okay, so what about a gene size duplication? So first of all, the mechanism that's going to give rise to gene size duplications are the unequal crossing over that I've described previously. Okay, so here's an example of a phenotypic consequence that would result. So this is in the fruit fly again, Drosophila melanogaster. So here's what a normal eye looks like. And there's a gene that contributes to eye formation. We call it the BAR gene. Okay, and the normal allele for the BAR gene is B superscript plus. So if you're homozygous for B superscript plus at the BAR gene, you have a normal eye. And what do I mean by normal? You can actually quantify the normal eye by, it's built from little subunits, these little facets, and there's a characteristic or average number that are found in a normal eye, about 780. So through unequal crossing over events, you can get gene duplications for the BAR gene. So now the duplicated chromosome is going to contain two tandemly arrayed copies of the BAR gene because of an unequal crossing over event. So if you have a fly that carries a duplicated version of that chromosome that harbors the BAR gene, then you have a normal chromosome that carries a single copy. What happens to the eye? Well, here it is over here. Here's the heterozygote for the duplication. And you can see the eye is about half the size of the original. So there's a phenotypic consequence that results because of that gene duplication. The eye becomes smaller. Eye development is compromised. Normal eye development is compromised. What if you're homozygous for the duplicated version of the chromosome of the BAR gene? Well, the eye gets even smaller. It's about 1/10 of the original size. And here it is down here. Okay, so once you have a pure breeding strain that is homozygous for the duplicated version of the BAR gene, you can actually get another unequal crossing over event in those individuals. So you can create a chromosome that has 3 tandemly arrayed copies of the BAR gene on it. And then you can, do genetic crosses so that you generate an individual that's heterozygous like this. So they have 3 copies of the BAR gene on one chromosome because of the gene duplication events, the unequal crossing over. Then you have a normal chromosome with only 1 copy of the BAR gene. And then the eye is even smaller. So that's kind of interesting in and of itself, in that in these individuals, you have 4 copies of the BAR gene. Here you have 4 copies of the BAR gene, but depending on how they're arranged relative to each other, you get different phenotypes. Okay? So this type of situation can also occur, when unequal crossing over events can give rise to blocks of genes that become duplicated instead of single genes, as I just demonstrated. Okay, so when you have blocks of genes that become duplicated, what are the consequences? Well, the best example, textbook example that we can use in this case again is in humans. So here's human chromosome 15, a normal one represented over here. So we've already talked about that. We talked about deletions, right? Prader-Willi and Angelman syndrome. So again, remember, the reciprocal product of unequal crossing over is a duplicated version of a chromosome. One copy through unequal crossing over produces a deleted version of a chromosome. The other reciprocal copy produces a duplicated version of the original chromosome. So again, this happens. There's something unusual about that region around the centromere on human chromosome 15, where you get this unequal crossing over occurring to produce deleted versions of the chromosome and duplicated versions of the chromosome. Okay, so in any event, so here's normal chromosome 15. They call it type 1, so normal healthy individuals carry that version of chromosome 15. There's a type 2 version of chromosome 15 that carries a duplication around the centromere. And in this case, the duplication encompasses maybe a handful of genes. I'm making up the number, but say 10 genes. Okay. So that creates a genetic imbalance. However, this type 2 duplicated version of chromosome 15 does not give rise to any phenotypic consequences. So that type of chromosome exists in living, viable adult human populations, but there's no consequence phenotypically or readily observable consequences. But there is a type 3 version of human chromosome 15 that is duplicated for a much larger region around that centromere. So instead of encompassing 10 genes, it might encompass a total of maybe 100 genes. Okay, so that creates an even greater genetic imbalance. And so what does that lead to? Well, it leads to all kinds of phenotypic abnormalities that are listed over here, smaller size, mental retardation, seizures, learning disabilities, scoliosis, etc. So again, the point is that when you have these duplications that encompass large numbers of genes, blocks of genes, there are phenotypic consequences that can result. Okay, so in all of these examples, whether they be deletions or duplications, I've given examples, all of which would be considered to be negative phenotypic consequences resulting. But actually, gene duplication events can have a positive outcome as well, and actually a very important positive outcome in terms of a driver of evolution and the ability to, through evolutionary processes, to create more complex life forms, more complex organisms. So, with modern day genetic analysis, it's pretty obvious is that there are many genes that are represented in the genomes of all different types of organisms, higher eukaryotic organisms, for which we carry many copies of those genes, some of which are tandemly arrayed. As I've described previously, that tandem array is generated through unequal crossing over. But in other instances, the genes are distributed on different locations on different chromosomes throughout the genome. But in any event, the idea is that there are many genes that are represented within the genomes of higher eukaryotes for which we have multiple copies. So where did the multiple copies come from? Well, they came through gene duplication events. And if they're originally arrayed tandemly on a chromosome through, all kinds of genomic reorganizations, those genes can be scattered onto different chromosomes. So this process of gene duplication leads to, what we call gene redundancy, and that can actually be a very positive outcome. As I said, it can lead to the generation through evolutionary processes of more complex life forms. So I'll just give you one, what I think is a good example of that, and it's the idea that in most higher eukaryotic organisms, what you find is that ribosomal RNA genes, rDNA genes, are highly repetitive. Multiple copies exist within the genome. So why would that be? Well, what these ribosomal RNA genes do is that they produce molecules, they produce RNA molecules that become incorporated into a functional ribosome. And again, the ribosome is essential to life as we understand it, as we know it. It's the ribosome that allows for protein synthesis or protein production. So within a cell, it would make sense, if you're going to be using genetic information to make proteins, the more efficient you are at making proteins, the better off you're going to be, the better able you're going to be to utilize all of the existing genetic information within that cell. So a large population of ribosomes is going to be advantageous. So how can you make lots of ribosomes? Well, you need lots of ribosomal RNA genes to construct those large numbers of ribosomes, therefore making you more efficient at protein synthesis and making you more efficient at maintaining an individual cell in a living state. So again, over long periods of time, again with the application of various types of evolutionary pressures and forces, is that you can create more complex life forms due to the greater efficiency of protein synthesis. Okay, so that's the general idea as to where gene duplication events can create suites of genes within an organism's genome that can then provide advantage to the individual and allow for the generation of newer, more complex life forms. Okay, so I'm going to stop at this point. What I've described so far are two different types of chromosome structural abnormalities that are somewhat similar to the aneuploids and polyploids. We talked about the deletion as being a partial aneuploid, so you should expect phenotypic consequences resulting when you have your heterozygous for a deleted version of a chromosome. Duplications, again, would be similar. Duplicated chromosomes would be similar to a polyploid state where you have extra copies of entire chromosomes or entire haploid sets of chromosomes. But we still need to talk about other types of chromosome structural abnormalities, the inversions, and the two different types of translocations that I described at the beginning of this lecture. So we'll talk about those next time.